Prof Na Cai. It's my great pleasure to introduce Na. I have to say, maybe by the way of explanation, that Na already gave an inaugural introduction of her research at ETHZ in Zurich. So, which was also streamed by Zoom into Basel. So for us, it's the second inaugural lecture, but nevertheless, very much looking forward to it. Let me say a few highlights, or let me cite a few highlights from Na's career. Na did a bachelor and a master in natural sciences at the University of Cambridge before she continued with a DPhil in clinical medicine at the Wellcome Trust for Human Genetics at the University of Oxford with the supervision of Jonathan Lind. She did a postdoc at EBI, the Sanger Center, in the UK from 2016 to 2019, and then started her own group as a PI from October 2019 to January 2025 at the Helmholtz Munich campus at Helmholtz Munich in Germany, where we had the luck to convince Na that BSSE and BRCCH would be the place to go for the future. Let me cite a few awards that she had, which she was successful of securing. There are the Friedmund Neumann Prize from the Schilling Foundation in 2023, the Early Excellent Science Award in Biology from the Biofoundation, the Theodor Reich Early Career Award from the International Society of Psychiatric Genetics. So she comes with a lot of laurels already to the BSSE. Of course, Na's role here at BSSE is very special. She is one of the professorships that the Botnar Foundation has made possible for BRCCH. This is one of the two who's arriving at BSSE, and we're very happy to see Na and her future colleague to flourish here in the department for the benefit of BRCCH and child health. Maybe just a few words to those who are not from BRCCH. Because this year, we are here at BSSE, so maybe let me quickly cite a couple of things. BRCCH's mission is to embrace the expertise of our partner institutions and foster a research community that is able to develop new healthcare solutions for the benefit of young people worldwide. And so for the Botnar Foundation itself is a philanthropic foundation founded in 2023 by Marcella Botnar in memory of her late husband Octave Botnar and her late daughter Camelia Botnar. BRCCH itself started 2017, was officially founded in 2019 with a contribution of 100 million Swiss francs, and is currently operating under the supervision of Georg Hollander and Daniel. Is correct? Yeah. So I'm very much looking forward to the talk by now, and I give you the floor.
Thank you. Thank you, Sven. Is my mic on? Okay, lovely. Thank you all for coming to my inaugural lecture at the BRCCH. And thank you, Sven, for the really lovely introduction. I'll get on to my talk straight away. I'm a statistical geneticist, and my work focuses on the genetics of depression. So what I do is I use statistical methods to test for the relationship between genotypes and the variation in genotypes between people and their phenotypic variations. And for phenotypic variations, in my particular case, would be people's liability to get mental health diseases such as depression. So let's talk about depression for a bit. Depression is one of the most common and relatable diseases in modern times. As many as 1 in 5 people will get depression in their lifetimes. If you're a woman, especially near the period of childbirth, you are more at risk than usual. And also it causes suicide in many times in younger people. This is the current clinical criteria for diagnosis of depression. As you can see, it is a big laundry list of symptoms, and which the clinician would assess in the patients through a detailed clinical interview. So none of these symptoms we can see on this list currently matches anything that is quantitative or biological. You can't measure anything such as fatigue or loss of energy or worthlessness, etc. So they don't map very nicely onto biological pathways as we normally think about. In other words, these symptoms are thus seen as indicators of an underlying disorder or underlying disease, rather than a good description of the biology of the disease itself. So this is what's causing our bottleneck right now. We don't know the nature of the disease, despite knowing how it looks like in people. There is no biomarker for objective diagnosis, and as a result, there's high misdiagnosis rates in the clinic. There's also no reasonable way to make a targeted treatment. Basically, what happens now is treatments are pretty much trial and error, and 50% of people don't respond to their first treatment and have to go through several in order to see any kind of betterment. So what we want to do as geneticists really is to use genetics as a tool to understand the nature of this disease, such that we might be able to get to actual molecular targets that we can use for people's treatment. And therefore, we can help people, many people who suffer from this disease nowadays. So the first question we have to ask in order for all of this to be valid is depression genetic? And this is something that I get asked pretty much. This is the first question I get asked every single time I talk to somebody that, I work on genetics of depression. Because it's so easy to think of depression as something that you normally get when, for example, something bad happens to you. As researchers, we get a bad reviewer's comment, we get a grant rejected, you get, sit in bed for a bit worrying that you're not very worthy, etc. So it's very easy to think of depression as something that is, just what happens to us on a normal day. So what they are actually asking by asking whether depression is genetic is they're asking whether it's heritable. So whether there are DNA elements, genetic variations in people that can be passed on to their offsprings that would decide whether this person has a greater risk for getting depression in their lives than others. So we are going to call this heritability, and heritability really is the proportion of the total phenotypic variance. By phenotypic variation, I mean the liability to getting depression that is heritable, that you inherit from your parents, as opposed to all of the things that are not heritable. So things that are heritable are things like genetic variation. You definitely get 50% from mom and 50% from dad. Things that are not heritable are things like winning the lottery. That doesn't run in families. Although the interesting thing is that there's this thing called common environment. So although winning the lottery can't run in families, the habit of buying the lottery ticket might run in families. And that is what we would call a common environment. So common environment encompasses other things such as wealth, healthcare access. These are the things that run in families and are confounded with genetic factors that also run in families in a genetic study. So very often, things like common environment can also be detected in a genetic study along with genetic variations. And this is something we need to take note of when running our genetic studies. So some of the models explicitly control for some of these common environments when performing the estimates for what is heritability of a disease. For example, some of the earliest quantifications of heritabilities of depression were done on twins. We know that monozygotic twins share 100% of their genomes, and dizygotic twins share 50% of their genomes. So we find a lot of monozygotic twins, we line them up in pairs, and we find a lot of dizygotic twins and also do the same and line them up in pairs. We would see that the rate of having a disease could correlate between the pairs of monozygotic twins and the dizygotic twins. And you can get the respective correlations. As we can see in this toy example, the monozygotic twin correlation is 0.6, that of dizygotic twins is 0.2. And given the fact that dizygotic twins share only half of their genome as opposed to 100%, we can use all of these information to calculate how much of this correlation between twins can be due to their sharing of genetic factors. And the equation is shown on the slide. Sorry, I'm not very used to this pointer thing. The equations are shown on the slide. Better not use this anymore. Equations are shown on the slide, and you can see there's a C term there for the common environment, and they are cancelled out once you actually have the twin heritability equation. So this method, notably, assumes that dizygotic twins and monozygotic twins share the same kind of common environments. That's why you can cancel both C's out. And this is sort of like the basis of a lot of the twin estimates that we see on the following slide. So this is a plot from a paper that meta-analyzed several twin studies on depression. And what it shows is that the heritability of depression is around 40%. So yes, it seems that depression is heritable, and there is some, reason to study it in terms of genetics. Now, all of these twin studies are done prior to the sequencing of the human genome, the large-scale genotyping of a lot of individuals. So this is, and it's mostly done before I entered the field at all. So in 2011, when I started my PhD in Oxford University in Jonathan Flynn's group, we are no longer doing twin studies. What Jonathan and his collaborator Ken Kendler has collected is a lot of depression cases, actually 6,000 depression cases and 6,000 controls. And what they've done is that they've genotyped everyone at millions of variant sites in the genome. And the goal is to perform what we call the genome-wide association study that tests each of these millions of genetic variants against whether one has depression or not. So I performed the first GWAS on depression, as shown on this slide. On the x-axis, you have the position of the genetic variant along the genome. On the y-axis, you have the negative log 10 p-value of the statistical association. The higher the value on the y-axis, the more statistically significant it is. And the red line here is the significance threshold. So because we're testing millions of tests, we can't use the significance threshold 0.05 anymore. We have to do multiple testing correction. And everything that is above that red line, the horizontal red line, is what you normally hear people call a GWAS hit or peak. And this is what we mean when we say we have identified 2 GWAS signal for depression in this paper. These are the first ever GWAS hits identified for depression, and these results are from 2015. In the following years, the field of quantitative genetics, which is the field that does GWAS, has experienced the era of large data. So what happens is that GWAS was performed no longer on thousands of individuals, as I've shown on the previous slide, but on tens of thousands or even hundreds of thousands of individuals. And as a result, the number of GWAS hits also grew from 17 to 102. And in the latest big GWAS meta-analysis performed and just published earlier this year, we are now in the times of performing GWAS on millions of individuals and finding altogether 636 genetic associations with depression. So this is great progress in many sense. In a span of 10 years, we've gone from 2 GWAS hits on depression to 600. And in theory, now armed with 600 genetic associations of depression, we should be well situated to infer a huge amount of biology about the nature of depression. So right now seems to be just about the time where we can, progress in leaps and bounds, and there's nothing stopping us now. We've got all we need. But in fact, I can finish my presentation now if that's the case, but that's not the case. So there are 2 real issues with this statement that I've made just now. And the first is that depression is actually heterogeneous. What we've done so far, what I've shown you so far, is performing genetic analysis on all depression cases put together in a massive pot. And we put all of them together, we get 600-ish GWAS hits. So the question is, if depression is actually heterogeneous, how do we now know which of these 600 hits are for which kinds of depression? And are we missing some just because we put everyone together? So then to answer this question, we first need to ask, are all depression cases the same? Well, if they're all the same, then we don't need to worry about this issue. So there are many, many studies on whether depression is heterogeneous. If we look at the literature in psychiatry, in psychology, in genetic epidemiology, there are literally piles and piles of this. And we, of course, wanted to add to the pile, and we've done this using statistical genetics. So this is our attempt at really, answering this question from the statistical angle. So we're saying, if all of the genetic alleles, genetic risk alleles that we found associated with depression were for a single kind of depression, then we would expect all of them to be pretty much additive to with each other. They all should act leading to one form of depression. If that's the case, then we would expect any random selection of these risk alleles to be equally likely to co-occur in a group of depression patients as any other set of risk alleles. Yeah, we should be able to test this statistically as well to prove that this is true. Now, if depression is in fact actually two diseases or actually two subtypes that are led to by different genetic risk variants, or two or more, doesn't matter whether it's two or more, then what we would see is that not any random selection of risk alleles would be equally likely to co-occur in a group of patients. Rather, those risk alleles that lead to the same kind of depression would always co-occur, and those that lead to different kinds of depression would not co-occur in the same group of people. Yeah, and we should be able to test this statistically as well. So using this approach, we are able to statistically prove that depression is a heterogeneous disease. Now, this method doesn't help us solve the issue of which of the 600 GWAS hits that you saw just now fall into which group. That hasn't been solved yet, and that is still what we are trying to work on. So we're not the only people working on this. The fact that depression is heterogeneous has been always observed across clinical history of depression. So people have seen that depressed cases have different symptom profiles, they have different ages of onset, they have different recurrence, and some people have depression only when they are close to their childbirth. So these have all been sort of already classified as clinical subtypes of depression, and genetic studies have already been performed on these different clinical profiles of depression. And they do find that these different types of depression are all genetically different from each other. But I would argue that being genetically different from each other is too low a bar. Almost anything is genetically different from each other, except if you arrive at a perfect subset of depression, right? And this is the problem that we are trying to solve right now with some of the analysis in our group right now. So we claim that clinical profiles do not correspond neatly to biological mechanisms. If we classify people by clinical profiles, which I've said just now, don't map on nicely to any biological pathways, then just identifying people with a symptom versus without a symptom does not mean that you've identified people with a mechanism versus people without a mechanism. You cannot make the equation that way. So as a result, we cannot just classify people based on their clinical profiles, do genetics on the two groups, and hope that they will give us the right biological mechanisms. If we can't trust clinical symptoms or profiles, what can we go on? The thing that we really want to start doing now is we want to interrogate molecular measures. Now, ideally, we would get tissue-specific epigenetics, gene expression, neuroimaging, metabolites. We might also get drug responses from all of these different people. Let's stack all of the molecular information we can for all of the depression patients. And what we want to do then is to try to cluster these depression patients into groups that have the similar metabolic profile. Or molecular profile. There are many problems with this approach too. The first problem is that there is very prone to artifacts. So all of these molecular measures, for example, carry with them quite a lot of biases depending on how they are quantified, who they are quantified in, and how they vary in the normal human population in the first place. So just by clustering them into groups of people, you can't necessarily identify the right groups of depression cases. So what my postdoc here, Zheng, who is in the audience, is doing right now is he is performing what we call contrastive learning on molecular measures that are available in cases and controls. So as opposed to just clustering molecular measures in cases using clustering approaches such as PCA, what we do is that we use contrastive learning to identify those axes of variation that are common between cases and controls, and then we remove them before we identify those axes of variation that are most prominent only within cases. In this case, we will remove a lot of the artifactual variation in the data set and only leave those that are potentially more relevant to disease cell typing. We're still investigating this approach right now. The other problem, I said there were two problems with the molecular approach. The other approach, as you can see on the slide, is that this is too wishful, right? How am I ever going to get epigenetics, gene expression, neuroimaging, et cetera, in hundreds of thousands of people, as I've said just now? That's almost impossible. It's going to cost me too much money. Definitely can't get that. But what we could do is to predict them using state-of-the-art sequence-to-function models. Now, there are a huge amount of models out there. Literally every day, there's 2 papers appearing on bioRxiv telling us how to do it. So what we want to do is to explore a few of them, especially the ones that are most prominent in the field, to see whether we can predict all of the molecular layers in the people we have genetic data on, so that we are able to get to the molecular profiles, even though if it's an imperfect one, to see whether we can start classifying people this way. So this is also very much work ongoing right now. Now, I've said a lot of things about depression being heterogeneous, and that's one of the issues about identifying 600 GWAS hits for depression, but yet still not getting any biological insights. There's actually another issue with these 600 genetic associations of depression, and that is in the input data that went into the GWAS that helped us identify all these 600 variants associated with depression, only 4% of them came from clinical interviews. 54% came from electronic health records, 14% from self-administered questionnaires, and 27% from self-reports. So huge heterogeneity in data input into the genetic analysis as well. So then that leads everyone who asked me about depression, and for me to also ask the field, is clinical depression the same thing as self-reported depression? Are they so similar that we can put them together in the same GWAS meta-analysis? So I've actually done an analysis a few years ago to look into this. And on the plot here, I show you the heritability of clinical depression on the left in purple and self-report depression on the right in green. And in the middle, the blue bar, the light blue bar is depression based on electronic health records. And what you can see is that the heritability of all of these different types of depression are quite different from each other. The clinical depression has much higher heritability than the self-report, for example. And this really means that their genetic contributions to these different phenotypes of depression are not the same. So we shouldn't be expecting to get similar genetic results out of them. Now, it is now then questionable whether we should meta-analyze them in the same big study. So not only are they different in terms of heritability, they're also different potentially in how they are able to predict depression in a new person given their genetic data. So this is polygenic risk score on the slide now, and it is a usual statistical approach to predict a person's liability to getting a disease using that person's genetic data, as well as effects at genetic variants that has been previously identified in the different GWAS. So we can use a PRS, or for example, self-reported depression, a genome-wide association study performed on self-reported depression, to predict depression in a new person where we have genetic data. And it will give you some prediction R-squared. You can also use it to predict a non-depression phenotype, and you get a non-depression R-squared for the same PRS. And what we really want is for the depression prediction R-squared to be much higher than the non-depression R-squared, right? If we're predicting depression using a depression GWAS, it better predict depression best as opposed to some other things. If it predicts the top phenotype better than depression, then we haven't done a very good job at identifying genetic effects for depression. So we've derived this very simple new metric called PRS pleiotropy, which is really just the ratio between the two R-squared. The lower the ratio, the more specific our genetic polygenic risk score is for depression. And we can use this ratio to assess the different GWASs that we've seen that has been used in a big meta-analysis. So what we found using PRS pleiotropy is that it is low for depression defined using clinical criteria, which is the black line you see on the bottommost of this plot, than self-reported depression, which is shown in the pink and the gray lines in this plot. So as a, in summary, what I've shown you just now is that self-reported depression is much less specific at identifying genetic associations of depression as opposed to clinical depression. So yeah, this is an ongoing problem, isn't it? Because nowadays, our biggest amount of data has come from electronic health records and self-reports. We cannot avoid this issue. If we all go back to having only clinical diagnosis of depression, we will go back to the era where we only have thousands of samples. We cannot make use of all of this much bigger data sets that are collected using different means. So then the next question that our group answered over the past number of years is, how do we leverage all the types of data to study clinical depression? We don't want to throw away hundreds of thousands or millions of individuals worth of data. So the first thing that we did was we took this sort of easy question and asked, can we think about filling in the missing data for clinical definitions of depression in a big biobank where we have lots of information on self-reported depression, mental health questionnaires, socioeconomic measures, etc. So all of these other things that are related to clinical depression, but not actually clinical depression. I'll tell you why this is the easier problem a bit later on. But this is the easy version of the problem. So all we need to do is, in a big matrix of people where the deep phenotype of depression or the clinical phenotype of depression is missing, can we use the rest of the data to fill it in? So the first method we used is an off-the-shelf singular value decomposition method, that just factorizes the big phenotypic matrix into factors and weights, and then predicts it by combining those factors and weights and giving it some noise. We also developed a new deep learning-based approach for the same task. And our results are very promising. I don't have the results here on slides for you, but they do get us more GWAS hits. They are also quite specific to depression, much more specific than self-reported depression. So we are able to rescue some of the data in these big biobanks by filling in the clinical phenotype that we want, leveraging the rest of the information. Now I want to talk about the more difficult task of working with electronic health records. So unlike big biobanks and using questionnaire data where you have this nice table of columns and where rows are individuals and columns are phenotypes that has been collected on the same individuals, electronic health records are much more messy. So in fact, in each individual in the electronic health record look a bit like this. It's a sequence of diagnostic events that has happened in their lifetime. And these sequence of diagnostic events are sparse, they're irregular, they don't all happen at the same time. And each actual entry of a diagnostic code may be a true diagnosis of a person who has the disease, maybe a false positive diagnosis of somebody who doesn't have the disease because of misdiagnosis. And each lack of an entry of a diagnostic code could be because the person never came into the clinic but actually have the disease, and therefore underdiagnosis, or actually just because this person doesn't have the disease. So all sorts of possibilities there. In other words, what I'm saying is that I don't expect the electronic health record to be correct, because we know actually through many previous studies that the electronic health records contain quite a lot of different kinds of biases. So what my postdoc Ya Zheng has started doing is he's realized that rather than to take EHR sequences, diagnostic sequences, as the gold standard for predicting, for example, the next episode of depression based on the previous episodes of depression, he needs to integrate the EHR diagnostic codes with disease-relevant measures, either from self-reports, from questionnaires, from biomarkers, from tests, for example, that has been performed on these people. And by integrating them through a deep learning network, he's able to predict a lifetime disease liability that is potentially a much better measure of the disease than any single electronic health record code. And he will know how good they are by testing them against gold standard diagnosis that is available in very small numbers of individuals. So we've done this analysis on 9 different diseases, including depression, and depression is right here. Oh, you can't see people online can't see my cursor, including depression and generalized anxiety disorder. And what we've shown is that our predictions, which are in red and in blue, always do better at predicting at their correlations with the gold standard diagnosis than the electronic health record itself, which is in green. And the electronic health record itself, the raw codes from electronic health records, is surprisingly bad at capturing what the gold standard diagnosis is of all of these diseases. So we are continuing with this project, and there's a lot of other investigations we're doing, including how do we select the number of people to call back for obtaining these gold standard diagnoses to enable such a system as shown on the slide here. So please keep your eye out on Yajing's paper in the next number of months. So since we're on the topic of sequential electronic health record data, it is naturally it's natural to ask a very related question, and that is whether we can derive biologically meaningful disease trajectories from individual sequences of diagnosis. We often say depression don't happen in a vacuum. In fact, we observe that depression happens a lot in people with chronic diseases, and other diseases may lead to the onset of depression later in life. But all of this is anecdotal and empirical. So can we actually model it in a way that we can use for genetic analysis to really see whether there are different types of depression that are, for example, dependent on other comorbid diseases? So there's already quite a lot of work that exists on identifying disease trajectories. And one of them that is pretty prominent in the genetics field is to treat each diagnosis in a sequence of diagnosis as a word, and using natural language processing methods such as topic modeling to cluster these words into topics, and based on how often they occur together. So if we are able to classify individuals by the kinds of disease trajectories they have centered on depression, then we should be able to obtain depression subtypes in an orthogonal way from those that I've described using molecular approaches a few slides ago. So we are currently working with a collaborator group in Mass General Hospital on deriving depression-centric disease trajectories using diagnostic codes available in the UK Biobank. So in this data set, we have 53,000 people who have at least one depression diagnostic code from any medical records, including primary care and inpatient. And 14,000 of them have 20 years of diagnostic data on either sides of their depression diagnosis. So we use these 14,000 people for all of our analysis. Through centering the electronic health record on depression and restricting it to 20 years, what we're able to get is a matrix of individuals by codes that they get in this 20 years timeline, and where each entry is the number of times they get each of the diagnostic codes apart from depression. We then can use a topic modeling approach, such as Slayton-Dirichlet allocation, to group some of these codes into topics, and then to cluster the individuals on the topics that they have based on their codes loadings onto the individual topics. So one notable thing about this approach is that LDA doesn't take into account the sequence of events. So it doesn't know whether this code has happened before depression or after depression. So in order to distinguish those codes before and after depression and what's happening during the episode of depression, we've added before, after, and during to those codes such that they can identify those as different words. So using this approach, we've identified 5 clusters of individuals corresponding to 5 different disease trajectories for depression based on, for example, the number of diagnoses, their ages of onset, and other codes that they have in their trajectories. And to show you how some of these trajectories look like, trajectory 3, for example, as shown on the slide here, is characterized by having a lot of immune diagnosis after the depression diagnosis. And trajectory 4 is actually very similar to trajectory 3. They have similar numbers of immune diagnosis after depression, but they also have a lot of some immune diagnosis just before depression as well. So we identify these as different disease trajectories. Now, the question is whether these disease trajectories have any different, diagnosis and outcomes for depression. So we tried to do this by anchoring our outcomes on antidepressant use. So what we find that trajectories 1 and 2 show low antidepressant use across all classes of antidepressants. Trajectories 3 and 4 clearly use much more antidepressants than the rest. And trajectory 5 is interesting because it is specifically enriched for those people taking NDRIs, which are a form of antidepressants, but they are second-line treatments for depression and are much more commonly used for ADHD and narcolepsy, for example. So we can see that all of these disease trajectories that we found are different in terms of the antidepressants that they were given, at least by their physicians. We therefore asked, because especially trajectory 5 is enriched in people with taking drugs that are commonly given for ADHD, whether these people have a higher genetic risk for ADHD in the first place or other psychiatric diseases. And what you can see, so here on the left, we have labeled trajectories 1 to 5 and their genetic profiles for a lot of different diseases. And trajectory 5 has increased risk for quite a lot of cardiometabolic phenotypes, as well as alcohol use disorder and ADHD. So this is a massive contrast to trajectory 1, which have a depleted risk for all of the cardiovascular diseases, but a potential high risk for schizophrenia. So it seems that our trajectories are really able to tell different types of depression apart. And to summarize all of the findings that I've shown you, we found 5 trajectories over 20 years time point by clustering disease codes from the electronic health record. And we are able to find these trajectories seem meaningful because of their antidepressant use and also their polygenic risk for other disorders such as ADHD. So these are some of the current ongoing work that we have in terms of using electronic health records, and something that we are very interested in applying to, for example, younger individuals through, for example, BRCCH efforts. So before I end my talk, I do want to bring up the issue of depression. Is depression in adults and adolescents the same genetically? And this may be particularly interesting to the BRCCH audience. So it has been known for a long time that depression has an early onset group and a late onset group. And the early onset group usually have their onsets before 20 years of age, depending on which study actually investigates this. So this is the latest genetic analysis on early and late onset depression. By early onset depression, it is they mean people who have it before 25 years of age. And by late onset depression, they mean people who have it after 50 years of age. And they performed GWAS on both these groups of people, and they found different genetics leading to both of these kinds of depression. They also find that early onset depression have higher heritability than late onset depression, which is very consistent with many previous studies on this matter. Not only do they find different and higher genetic load for early onset depression, they also find that early onset depression have higher suicide risk in terms of both their genetic correlation with having a suicidal behavior or suicidal intent, as well as the actual risk for having a suicide attempt in the number of days that follow their depression diagnosis. So this is of particular interest, I think, maybe for all of the people who care about depression and suicide risk in young people. And what this tells us is that we might really need to have a better follow-up of people's depression symptoms and diagnosis and their outcomes once we've diagnosed them in the clinic. Not only do they have immediate sort of potential clinical impact on identifying and following people with depression that for their suicide risk, they've also found that early onset and late onset depression have similar enrichments in adult neurons in terms of their methylation patterns, but they have different methylation enrichments in fetal biomarkers, methylation markers. And this is very interesting because we've seen similar kinds of enrichments of fetal molecular markers for schizophrenia. So this sort of like, brings up another topic that might be of interest to the clinical crowd. And that is, are childhood depression or adolescent depression much more likely misdiagnosed because the individuals actually have a different psychiatric disease, but they were diagnosed as depression because the children or the adolescents couldn't yet well express what it is that they're having. So there are a lot of evidence for this. For example, early onset depression are able to genetically predict bipolar disorder a huge amount better than late onset depression. So the first two bar plots that you see in this slide, the two on the left, show the prediction of bipolar disorder by early onset depression given different kinds of genetic risk scores. And the right two bar plots show the predictions in late onset depression. And you can see that the late onset ones are totally flat, while the early onset ones give much higher and better predictions. The same is true for schizophrenia. So early onset depressions predict schizophrenia so much better than late onset depression. And it turns out it's exactly the same for ADHD. So this is another study that represented these data in a different way, but we also see that early onset depression are able to predict schizophrenia and ADHD much better than late onset. So there is a big concern of potential misdiagnosis. And why do we have this concern of misdiagnosis? In addition to the fact that children or adolescents may be less able to express themselves. This is a terrible slide. I'm sorry to show it to you this way, but I'm chairing a heterogeneity subgroup in the Psychiatric Genomics Consortium, which is an international group of scientists coming together to work on genetics of psychiatric diseases. And what is shown on the slide here is the different diagnostic instruments used on different sets of people. So in the leftmost column of this slide, I have the diagnostic instrument used for children and is meant to be used by their parents. So all of the questions says, does your child have 2 weeks of this symptom? And does your child have 2 weeks of that symptom, etc. The rest of the columns are adult questionnaires administered to adults, either clinically or through self-administration. And what the colors are items of these questionnaires that sound similar to each other just through looking at wording, right? And as you can see, the children's questionnaire, they do have items that look just wording-wise much like the adult questionnaire. But when you perform factor analysis, either on the phenotypic or the genotypic level, on the variations detected by these questionnaires, you will see that individuals, we don't identify clusters based on the colors you see on this table, by what kind of symptoms that the questionnaires are assessing, but rather by questionnaires themselves. So the context in which you ask the question is important, and who you ask the questions to is also important. Asking a parent may be very different from asking the child or adolescent themselves. So that is one issue that we're still trying to solve in this big international consortium. And this is the final slide that final science slide that I want to talk about. And that is how might age matter biologically. So we think of age of onset as a really weird thing, right? You could think of people with a more severe genetic liability to the disease to be much more likely to get it earlier. For example, if let's say, environment is random, my disease, whether I get the disease is a combination of my genetic and environmental liability. So the greater my genetic liability, the smaller my environmental liability needs to be for me to get the disease. The sooner I'm going to meet it in my life, the sooner I get the disease, right? This is one model. And another model is where age of onset is its own thing. It's biologically regulated because of, for example, physiological environments, neuronal development, connections, etc., and also potentially differences in resilience to stressors that you might encounter early in life. So we now wonder whether age of onset is its own thing biologically, which interacts with the genetic liability to getting the depression or any other illnesses, or whether it's just a proxy for environment and for how big your disease liability is. This is very much ongoing investigation in the group, and it may only be possible if we gather data on age of onset when people actually have their disease onset. And that's another thing I really want to work with clinicians in the context of BRCCH. So of course, there are many other questions regarding depression that I don't have time to go into today, regarding sex differences, treatment and outcomes, etc. But what I've told you about today, as well as all of these things that I don't have time to get into, really shows that this is very much work of a lifetime. We are trying to use genetic tools to assess and improve psychiatric phenotypes. We're trying to use molecular data to understand disease liability. And we are trying to use electronic health records to investigate disease comorbidities and trajectories and outcomes. And only with all of these efforts, we might have a hope of understanding the nature of depression such that we can derive drugs that treat it specifically so that we can help all of those people who currently have the disease. So I've come to the end of my presentation. I'd like to thank all of my collaborators, my group, all of my previous mentors, the biobanks and the data sets that I've been using to perform my research, NIH that has funded our EHR work, ETH Zurich, BSSE for hosting me here, and also, of course, BRCCH for allowing me to come to Basel. Thank you so much for your attention.
That was wonderful talk, and benefit of this in our lecture or first is that for this one we have time for questions.
Only easy ones, please. In
terms of mechanism, I know the genetics is the mechanism on mix, but what do you make of the striking association between bipolar schizophrenia and early onset depression? How much do they match together in the other way around? That is, is it in most cases people that will have schizophrenia will also get depression? And because my understanding is that in depression that there are no genes that give you a very high heritability. That is in contrast to the other ones. So could it be that the main driver for early onset are psychiatric conditions, unlike depression, and depression rides on top of that, and for the later ones it rides on top of other diseases. But it's very, very hard to pin down the causes of depression itself mechanistically.
I think you're totally right. And I think, these are the kinds of hypotheses that we are, for example, testing. So what you said, for example, that schizophrenia, bipolar disorder, many of these diseases that have much more of a developmental origin and much more of a, genes with large effects, may be the ones that are actually, happening first. Whether they drive depression or not, hard to say. We don't know for now. Maybe they are misdiagnosed as depression in some young people, especially ADHD. This is what I hear anecdotally from both clinicians and parents, that their children are miserable because they actually have ADHD. ADHD is a relatively recent psychiatric diagnosis to be included in the clinical definitions. So prior to that, there really is no diagnosis to go for, except the children are miserable. And that's why many of them may be given other codes if you look at electronic health records from a very long time ago. So it's hard to say whether these diseases give rise to depression, which is a different kind of thing, or they're misdiagnosed as depression. But I think, for example, going in to analysis with these kind of hypotheses flashed out with the aim of sort of like confirming or disproving these kind of hypotheses is potentially the way to go in the future.
Very interesting point, actually. So I was just thinking of the genetic overlap between schizophrenia and bipolar disorder, where you have an estimated 8,000 common variants contributing to schizophrenia and about 6,500 to bipolar disorder. and the overlap is extremely high. So you have only 200 something that are really specific to bipolar disorder. And of course, there's a lot of discussion around it. What is it in the end? Is it misdiagnosis, or are there really common SNPs that predispose to a more general liability, and then the specificity is provided through either specific genetic factors or environment or whatever? So that's an interesting question. So I want to congratulate you. Very interesting talk. We are working on bipolar disorder, so I know many of the problems that you or challenges that you were describing, although the heritability in bipolar disorder is somewhat higher than in major depression. But one thing was you showed the impressive number of ETHZ that there are in major depression at the moment. But I mean, I know it also from bipolar disorder. We have many hits now, but we don't have the true, let's say, causal variants at each locus, and we don't have a molecular mechanism that underlies each of these ETHZ. So this is a lot of work, obviously, but I was saying there's a lot of work to be done on these 600 hits. So we know, I mean, there are 600 hits, and maybe they're close to this and that gene, but it's in the end, for most of these hits, it's completely unclear what they are doing at the molecular level. And I think that would be also very important. And I know that there are groups embarking on this now, but it's a lot of work and a lot of very expensive work and time consuming and so on. But I think there's a lot to be learned from the hits that we have already now, right? So this is a very nice foundation that we have at the moment. And that was rather common. The question was, for you said you are applying this to children. So what would be the aim actually? So you have children that show up with certain symptoms already, and then you would sort of look at the polygenic risk and maybe other clinical symptoms to predict a disease trajectory, or in the first place to predict whether the child is prone to develop depression. I mean, which is, of course, we know that this is not easy to make predictions before the onset of disease. So that was what I did not get completely. Is it rather if you have a child or an adolescent with the first diagnosis or suspected diagnosis of depression, and then you say, okay, let's have a look in a research context. What's the polygenic risk score? What's the predicted disease trajectory, which may have implications for the clinical management of the patients? But it's not so easy. I was not completely clear what you mean by this.
Yes, I see. So I don't necessarily need to go into the clinic and actually meet children for this. There are a lot of data sets, for example, from the Nordic medical registries, where they have lifelong diagnostic information of almost everyone in their nation who choose to be part of the medical system, from birth until they are quite old. So this is probably where we will start. We're already working with colleagues in Denmark on this, and the hope is that we work with more data sets also. So here, what we are expecting to see is that if, let's say, childhood diagnosis of depression is more commonly a misdiagnosis than other forms of psychiatric disorders, you would more often see that these people switch to a different psychiatric diagnosis later on in life when they are better able to express, for example. This is a hypothesis. We haven't tested it, but there are, for example, our colleague who is now working with us on the trajectory data in Mass General. He has performed this analysis in a Colombian biobank in his previous work. And there we see that for different psychiatric diseases, the switching from and switching to is very different. So for example, for depression, there's a lot of instances where a person with original depression diagnosis end up later in life with some other diagnosis. But less of that is true for other diagnoses right now. So yeah, I think there is something to do there with existing data that has already been collected and the individuals that are in these data sets are probably now already 60, 70 years old, but we do have data on them for many, many years already. This is probably what we start with rather than just going to the clinic and asking for more data now. Yeah.
Okay. Maybe. You could stratify patients with depression based on which treatment, which drug they respond. Is there any evidence that this segregates with genetic risk?
Yeah, there's quite a lot of studies on this. There are about, as of last year, I've had a student, a former student, look through the literature on the drug studies of on depression that are linked to genetics of any sort. And as of the end of last year, I think there are around 20-ish studies on this. And the problem there is, in most data sets, there's no real data on whether the person has responded to a drug or not. In very few data sets, they actually have a scale on whether the person responded to a drug, and that you can only administer to a small number of people. In an electronic health record setting, all you see is this person has taken drug A at time point A. They then went on to take drug A again at time point B, and then they stopped for whatever amount of time. Then they start taking drug B. It's like, okay, like, why did you stop taking drug A and start taking drug B? There is no explanation to this, right? So this is the limitation of the data that we mostly have. Electronic health records, unless coupled with clinicians' notes on the reason why this person has stopped taking drug A is because they cannot digest it properly, it causes digestive issues, or they're now not sleeping anymore. And, different side effects mean different things biologically. And if we have access to those clinicians' notes, for example, we can extract it with the LLM and everything to sort of like start modeling this in our analysis. But without access to this, it's very difficult to figure out why somebody has taken a drug and whether it's not working or it's actually working amazingly well. That the person has gotten better, so they stop taking it, right? So currently, I think it's still very early days for investigating drug and treatment outcomes. With better curation of data and better sort of like integrating clinicians' notes and the diagnostic and treatment data, we should be able to do this much better in the future.
Any question?
Thank you for the talk. It was very interesting. I have one question regarding how do you reduce causality given the confounding of the temporal nature of events? Let's say I have a bad gene for giving a good interferon response, and that leads to long COVID, and that leads to infection, right? So for you, is it confounding philosophically, or for you is it okay, if a bad interferon response gene leads to that kind of chronic disease that leads to depression every time, that is a good enough reason for you to say that this causes depression?
Yeah, I see what you're saying. That's a difficult question. I think I lean more towards confounding rather than treating it as part of the disease mechanism for depression itself. Because without, yeah, I think I lean more towards confounding, but this is a very difficult problem, I think. It's unavoidable. I mean, everybody has their disease trajectory. Maybe this is why our trajectory work is meaningful in a way that I haven't thought of before. Maybe one interesting thing for that, since we're having this discussion, literally just thought of it, so it's not a half-baked thought for sure, is whether that confounding affects everyone or just a few people, depression. A subset of individuals who would have depression in their lifetime, so literally all of the people who have genetic risk for depression at all. If it affects everyone with depression, then confounding, right? It's like there's no subset. But if there are people, there's a subgroup of depressed, people carrying depression risk alleles that are particularly susceptible to this interferon effect, for example, then it's hard to say. Then we have to start thinking about it as a potential thing about depression itself, right? Yeah.
Okay, thanks very much to Na and to everybody participating in the discussion. And I'm happy to say that if you're interested, you can continue with the discussion outside where there's an apéro.
prepared for everybody. So thank you very much for coming, and thank you very much for this wonderful talk.
Thank you.